Hello students, I hope you are doing well. In the last week we learned about different cellular components such as ribosomes, rough ER, smooth ER, Golgi apparatus, and how the protein is being processed in the vesicle and in the Golgi. And we also learned about lysosomes, etc. And in this week we will learn about the function of mitochondria, how the energy metabolized, and cytoskeleton and plant cells, chloroplast and cell wall, components of cell wall, etc., and difference between animal and plant cells. So mitochondria, we know that the energy ATP is produced in the mitochondria, and mitochondria is also known as the energy house or powerhouse. So basically they are involved in the production of the energy from glucose molecule. So like nucleus and other cellular membrane, the mitochondria is a double membrane. The outer membrane is smooth, and the inner membrane is inside the inner membrane there's cristae. So this is cristae or crista, plural is cristae. So the inner membrane involves in the oxidative phosphorylation, and inner membrane also called matrix. So cristae are called matrix, and the inner membrane contains a lot of enzymes that is required for the um, energy production, and in the inner membrane there is proton pump, that is hydrogen ion or proton. So we'll see that the function of this proton pump, and which creates concentration gradient or chemiosmotic gradient, which drives the oxidative phosphorylation. Oxphos is oxidative phosphorylation. We will see that in the next slide. So further, this chemiosmotic gradient and electron transport chain, which includes the process of oxidative phosphorylation, all of this produces more ATP. Also note that mitochondria also have their own DNA other than the cell's DNA inside the nucleus, but the mitochondria also have their separate DNA, a circular DNA, circular chromosome. So that is like a bacteria. And also the mitochondria produces its own ribosomes, that is 70S type. So it is also these two functions are quite like, characteristics are quite like bacteria like. And we also mentioned about the endosymbiosis theory, about how the mitochondria engulfed into the self, and once the mitochondria was a prokaryotic cell, independent, and somehow it got inside the eukaryotic cell. So this is the endosymbiosis. So the function is, we know that it is central role is to produce ATP through cellular respiration. But in the adrenal glands or in the gonads, there are mitochondria. They have different function like hormone synthesis. Also note that mitochondria is involved in intrinsic apoptotic pathway. So you might have not heard the apoptotic pathway. There are two types of apoptotic pathway. So at first, apoptotic or apoptosis means programmed cell death. So it's a natural process inside the cell. There are two pathway mainly of apoptotic pathway. That is, one is the extrinsic pathway. Another one is the intrinsic pathway. So extrinsic pathway requires a signal or ligand signal from outside of the cell, and in the cell membrane there are death receptors and fast receptor. Those are the receptors that trigger extrinsic apoptotic pathway. So you don't have to know that right now. You just remember that the intrinsic pathway, the other pathway of the apoptosis, that is mitochondria, that involves mitochondria. So when the cell get damaged, then mitochondria somehow. So there are another type of gene, pro-apoptotic gene, BCL2. So just know the name. So BCL2 genes get activated, and the mitochondrial outer membrane that leaks out, so that gets permeated, and it releases a lot of enzymes, and those enzymes activated caspase pathway. So caspase is the the it activates so many hydrolytic enzyme which further destroys or engulf the cell. So just remember the pathway, the mitochondrial or intrinsic pathway, apoptotic pathway. So moving to the next slide. So in this slide we will see the process of cellular respiration, how the glucose is metabolized and produces ATP. So in the cytosol, so you can see this is the cytosol that is outside of the mitochondria. In the cytosol there is glucose, sugar, glucose, 6-carbon glucose. Glucose is break down by the glycolysis into 2 pyruvate. So glucose is 6 carbon and it produces 2 pyruvates. Pyruvate is 3 carbon. So 2 3 carbon means actually it is like breakdown of the glucose into half 2 molecules. So glycolysis is a breakdown process of the glucose, and it also produces ATP, 2 ATP net. It produces 4 ATP, but 2 was consumed, so net 4 ATP. And it also produces NADH. So the NADH further oxidized to produce ATP. So and this NADH oxidizing enzyme resides in the inner membrane matrix of the mitochondria. So we will see that, but the pyruvate, it can enter into different cycles. So for example, it can go in the mitochondria. Actually it happens, the cell needs energy, and the most of the energy is produced within the mitochondria, within the oxidative phosphorylation. So in the mitochondria, as the pyruvate enters in the citric acid or Krebs cycle or TCA cycle, and it produces NADH and FADH2. So these are the molecules that mainly catalyzes the electron transport chain and the chemiosmotic gradient. We will see that. So NADH and FADH2, these are the mainly driver of the ATP production. So most of the ATP is produced by NADH and FADH2. So one pyruvate, when it finishes the TCA cycle, one pyruvate produces 3 NADH and 1 FADH2. So 2 pyruvate produces 6 NADH and 2 FADH2. So on average, 1 NADH produces 2.5 to 3 ATP. Actually 3 ATP, but we get 2.5, and 1 FADH2 produces 2 ATP. So after finishing the TCA cycle, 2 pyruvate produces 6 ATP and 2 FADH2. But please note that in between these two processes, the glycolysis and citric acid cycle, there is another step, that is preparatory step just before the citric acid cycle. So the pyruvate that the pyruvate itself get oxidized and also produce 2 NADH. So in total we can say that TCA cycle, including its preparatory phase, produces 8 NADH. So it is, I wrote it down here, 8 NADH and 2 FADH2. And previously also from the glycolysis there are 2 NADH. So this NADH2 and FADH2 further produces ATP in the process of oxidative phosphorylation. So oxidative phosphorylation, mainly there are enzymes and proton channels inside the inner membrane of the mitochondria, and when NADH and FADH2 enters this inner membrane, there are channels, proton channels. So there are also enzymes. So enzyme catalyzes proton release. So this proton release from the NADH and FADH2 produces a gradient, concentration gradient. Concentration gradient means when a membrane is involved, one side of the membrane has a higher concentration. If the other side of membrane has a lower concentration of the solutes, then there is a concentration gradient or concentration difference. So osmosis, the phenomena of osmosis means movement of the particles or the solutes between a semi-permeable membrane. So the movement should be from high concentration to the lower concentration. So that is osmosis. And chemiosmosis actually refers to those proton atoms that drives this osmosis process. So in the process of this electron transport chain, the NADH and FADH2 carries out this process, and ATP is also produced when the NADH FADH2 get reduced. So that is in a nutshell oxidative phosphorylation, and most of the ATPs are produced in this cycle, oxidative phosphorylation. So please note that if there is oxygen present, aerobic condition, most of the time there is oxygen present. So in the presence of oxygen, pyruvate enters in the aerobic respiration or Krebs cycle. So this whole process requires oxygen, and it can produce ultimately 36 to 38 ATP, 38 for eukaryotes and 36 for prokaryotes. This is in theory, but in practical we get less ATP due to different sort of cellular mechanisms or system loss, I guess. So in case of absence in the absence of oxygen, then the pyruvate enters in the different pathway that is called fermentation, that produces lactic acid. So this lactic acid or fermentation pathway of energy production is much more less efficient. That is quite inefficient. That is inefficient, and the oxidative phosphorylation or Krebs cycle inside the mitochondria, that is efficient, much more efficient than the fermentation process. So when a cell is under stress, and when a cell requires much more energy, but all the mitochondria are overloaded or saturated, all the enzymes are saturated, so energy is not getting that much energy is produced, but the cell requires energy demand is high. For example, in case of severe exercise or strenuous condition, so in that situation your muscle cells need much more energy, and also it is consumes a lot of energy, so there is high energy demand, but all your mitochondria are saturated and overloaded. So in that case, all those pyruvates enters fermentation process and produces lactic acid, and for that lactic acid the cells get tired too fast. So you can learn about the electron transport system or oxidative phosphorylation in details, but in a nutshell I mentioned in a simpler way. So the NADH and FADH2 gets reduced in this inner membrane and releases proton ion. And this proton drives the chemiosmosis, and in the process of this electron transport, the NADH FADH2 produces ATP. So here one caveat, that is, the ATP is supposed to be 36 to 38 in theory, but we get in practical, in the experiments we see that we get around 29 to 30 ATP. That is less than the theoretical ATP. So this is an ongoing research field, that why the ATP is not that much, or why there is less ATP than it should be. So is there any problem in the mitochondria, or how the ATP is leaked? ATP is not getting produced. So this is quite fascinating field. So in practical we get 29 to 30 ATP per 1 molecule of glucose. So going to the next slide. As I mentioned earlier, that mitochondria were thought to evolve from an engulfed bacteria. So it has characteristics like a bacteria. For example, the DNA that is circular, it has a separate DNA than the host eukaryotic cell. So it also has bacteria like ribosomes, 70S. So the theory it governs, that is, once the mitochondria were bacteria like, and somehow it got inside of the cell, that is eukaryotic cell, and through the process of endocytosis, it got inside the cell. So whenever endocytosis is involved, the host cell's plasma membrane form the vesicle. So here you can see the vesicle of the plasma membrane of the eukaryotic cell. But in the passage of time, somehow the vesicle from the eukaryotic cell that got lost. So the mitochondria, we see the double membrane of the mitochondria, actually the outer membrane is from the bacterial plasma membrane, and the inner membrane formed between the mitochondrial intercellular membrane bound organelles that form the inner membrane. So the engulfing or the endocytosis, plasma membrane from the eukaryotic cell that got lost. So the symbiosis means when the two species that got benefited mutually, that is called symbiosis, and endo refers to the endocytosis. So through the process of endocytosis, the bacteria was engulfed, and then through the process of symbiosis, both of the species got benefited and lived together and formed mitochondria. So this is the theory of endocytosis.